The community of cannabis cultivators has always been tight. We had to be. For most of our lives, we were persecuted, and in some jurisdictions, we still are. That persecution meant that we couldn't access many of the botanists we'd like to talk to, and that meant there were big parts of cannabis cultivation that was kind of in a biological black box. We couldn't see what was happening inside. All we knew was what we were doing and then what was coming out the other side. Now, though, between the creeping partial legalization we've got and the internet, we can now access the minds of scientists who support our efforts to grow magnificent, thriving, and medicinal cannabis plants. Today's one of those episodes where we get to look inside that black box. During today's episode, we will examine the relationship between the cannabis plant and the soil life and get a better understanding of how they talk to each other and how they work together for mutual benefit in this intimate relationship. If you want to learn about cannabis health, cultivation, and technique efficiently and with good cheer, I encourage you to subscribe to our newsletter. We'll send you new podcast episodes as they come out, delivered right to your inbox, along with commentary on a couple of the most important news items from the week and videos too. Don't rely on social media to let you know when a new episode is published. Sign up for the updates and make sure you don't miss an episode. Also, we are giving away very cool prizes to folks who are signed up to receive the newsletter. There's nothing else you need to do to win except receive that newsletter, so go to shapingfire.com to sign up for the newsletter and be entered into this month's and all future newsletter prize drawings. You are listening to Shaping Fire, and I'm your host, Shango Los. My guest today is plant biologist Sarah Lane. Sarah Lane is wrapping up her PhD in plant and cellular biology at University of Victoria. Her work primarily focuses on root exudates that are involved in iron uptake in plants and their potential medicinal benefits. Sarah consults for licensed cannabis cultivators in Canada and is an avid living soil cannabis homegrower. Due to her university research needs, she is also very experienced in fogponics and hydroponics. Sarah has a wicked large plant collection and is currently designing a specialty terrarium for growing cloud orchids. During the first set of today's episode, we will focus on what exudates are, their biological mechanics and how they interact with microbes and other residents of the rhizosphere. During the second set, we will talk about the relationship between exudates and fungal networks, signaling varieties of exudates, and address the challenges of a 24-hour light cycle. We finish up the third set discussing exudates and salt-based fertilizers, exudates and soilless growing mediums like coco coir, and the misconception of using unsulfured molasses to replace the nutrition that exudates provide. Welcome to Shaping Fire, Sarah.
Hey, it's great to be here. Thanks for having me.
Oh yeah, thank you so much for joining us and sharing your expertise. So let's jump right in. You know, I think that most people who cultivate cannabis, who have even heard of exudates, only really know that exudates are something that the cannabis plant gives off and that the microbes eat. And like, that's pretty much it. It's the food, it's the plant feeding the microbes, and that's about it. And, you know, we know that that's actually like overly simplistic and misses the big picture. So why don't we start by kind of getting everybody on the same page, and if you would, explain what exudates are.
Okay, so exudates are basically all of the different molecules and components that the plant makes in the roots that are like given off to the rhizosphere. So there's a bunch of stuff inside the plant cells and inside the root that happens, and then exudates are everything that the plant makes specifically to transport out into the soil. And there's lots of different types of exudates. They can be sugars, they can be amino acids, they can be specific types of secondary metabolites which are not used for nutrition, and there's many, many different functions of them as well.
So just to give us an idea of like how many different exudates there are we talking about like there's like 5 buckets or 50 buckets or 100 buckets? Like how many varieties are there just in general?
Oh goodness, that's a tough one. There honestly are so many different types of exudates, and we're just scratching the surface of what exudates actually are. Like off the top of my head, like functionally, there's probably like 20 different groups of exudates that I know of personally, but I think exudates are extremely hard to study because they are in the soil and the roots. So I think we'll probably keep finding new ones over time.
This is kind of, I get the idea now that this is kind of like asking you how many types of bacteria are there. There's just like a ton in nature, and we're constantly finding more as our technology gets more sensitive.
So true. Yep, that's definitely the case.
All right. So, like, to help illustrate what some of these exudates are, can you give us like 3 different examples of exudates that, you know, maybe you particularly like, just so that we can kind of wrap our head around, you know, what they do?
Yeah, sure. So, I mean, the ones that I work with specifically are called phenylpropanoids. They're actually sort of a group of molecules that come from the process of building up lignin, but they have lots of different uses. And those include things like some of the more complicated ones are chlorogenic acid, which is actually found really in high amounts in coffee, so people might be a little bit more familiar with it. There's also amino acids that not only are used for protein building, but plants can make special non-protein amino acids that are really useful in finding out things like fishing out iron out of the soil and that kind of thing. And then the other one is flavonoids. There's actually a bunch of different types of flavonoids, which is a big, huge supergroup of different types of molecules. And they're involved in everything from antioxidant behavior to even signaling to mycorrhizae and things like that.
All right. So you've mentioned, I think I've pulled out like 3 different categories from your examples. There would be like this, the only ones I was aware of, the nutrition, right? And then you've mentioned signaling a couple times, and then you sounds like there are some that actual process, like things like nitrogen and things like that. They're like more like kind of like worker bees. Is that, do all of exudates roughly fall into these 3 categories, or are there many more categories to like what they do?
I think functionally we can kind of bin them, if you want to call it that, into a couple different groups. There's definitely like interacting with nutrients directly, which is kind of where my expertise is. There's signaling back and forth, like talking to bacteria or fungi or that kind of thing, where they interact with the microbes and the microbes interact back. And then there's another big group that are almost like anti-herbicidal. Or herbicidal, sorry. And those ones are responsible for sort of clearing the way for the plant to grow. I'm sure there's more than that, but I think that's kind of like the big, huge groups.
So I think I have an overly simplistic understanding of the back and forth, because the way I normally think about the relationship with the plant and the microbes and the rest of the life in the rhizosphere is that during the daytime, the plant will be fed by the microbes and the rest of the players in the rhizosphere during the day. And then during the dark period, the plant will then turn around and send the required nutrition back into the rhizosphere. And so it's like a back and forth, like a tide thing, dependent on whether or not it is day or night cycle for the plant, which is kind of quarterbacking this back and forth, is how I generally think about it. But that may very well be way too simplistic. So will you kind of explain how it goes back and forth?
Yeah, so I think like the basic idea of signaling is a little bit like texting. It's not so much that the plants are providing nutrition directly to the roots, although from the roots to the microbes, although in some cases it is. And it's not always that the microbes are providing nutrition back to the plant. There's a lot of signaling that's going on. And that's kind of like, if you can imagine being at home and, hey, can you pick me up some milk from the grocery store? But it could also be like, hey, I've got cookies when you come home, and that kind of thing. So that's kind of what I mean by signaling. And that signaling happens all day, every day, at any time. So there is some regulation with light interacting. Obviously, the light drives photosynthesis and that kind of thing. And when the plant is on, it's often needing a lot of different types of activities like nutrient acquisition, and maybe it's doing some, stress responses to help against pests or something like that. And a lot of this happens in the daylight. I don't think we know enough about whether or not it happens in the dark, because the roots are always in the dark, technically.
Sure.
Does that make sense?
Yeah, it does. The way that you described it, this kind of like all these different microbes and protozoa and then the plant exudates all kind of calling out to each other and signaling, pick up milk, or I have cookies when you get home. And and if everybody is doing that, I suddenly got this idea of like this cacophony of signals back and forth, and like shopkeepers calling out what's on sale, and kind of like some kind of like big Turkish bazaar, and like everybody trying to, get everybody's attention. Is that a reasonable picture of what's going on?
It's a beautiful picture of what's going on. I really do think that's exactly what's happening, because we often think about it being just the plant and the microbes interacting with each other, but the microbes interact with each other too, and with fungi and with different organisms around. Like, it is just a huge web of different organisms talking to each other. And sometimes that is like, selling your chips on the, or the hot dog stand or whatever. And maybe it is advertising for sale. And it's very, very, very complicated. And it's something that I think we wish we knew more about, but trying to dissect what each of those components is contributing to the soil life, or even just how the plant is growing in the soil, is really complicated. I like that though.
Yeah, and it's strange too, because like I've always kind of thought of the soil as, while a violent place often, pretty calm actually. Like it's kind of serene in there, but it's probably because of the level of magnitude. I'm living on this human level of magnitude, but if I could see really closely under a microscope, I'd be seeing all this activity in the rhizosphere, and so it's only quiet to me because like I can't hear it.
Absolutely. It is very barely controlled chaos down there, let me tell you.
So do we have any technology or any way to gauge the amount of, rambunctious signaling that's going back and forth? Like is there any kind of like anything that we can like put into the soil to gauge signaling that's taking place in the soil?
I think it's pretty complicated, but there's some interesting things that go on. So I guess it depends on what level. So what we're talking about right now is on the chemical level. And really the way that a lot of people are doing this is either bulk. So they take a big sample of soil and then measure it for different types of organisms or different types of signals that they're producing. But then trying to tease that out, we can tell differences, like maybe between different types of soil or different types of communities, different types of plants, that kind of thing. But I think it'd be really hard to pick out which one is responsible for exactly which one. And so the other option is to take an individual plant and an individual organism and see what they do together, just the two of them. And it's one of the real challenges with like soil ecology or like ecology in general, is just trying to pull out, you either have to treat it as a whole and just accept that you don't understand the individual relationships, or you have to get in there and try and tease them apart one by one. Does that make sense?
It does. So let's talk a little bit about a couple of these different varieties. So the one that we've talked about the most so far are the signaling. And so we understand what they're trying to signal, like I have this type of nutrition, or maybe I need this type of nutrition. How are they doing that? Are they doing that primarily chemically, or is it an electrical charge, or what is at that very small level? What is the mode of communication?
It is chemical. I mean, for the most part, it is like plants. If we go back to the exudate thing, like these plants are making these very highly specific molecules. They're basically, in some cases, like an invitation. And then those are actually like taken up by the microbe in question. And that changes what the microbe can make. So it actually interacts after a long signal in cascade inside the cell. It'll interact on the genome, and then they'll be able to produce different types of molecules based on that information. And then that ends up getting made and exuded out by the microbe, and then maybe that's an RSVP response card back to the plant, and then they kind of do this back and forth. Or sometimes it might just be like they actually make a different molecule, and that molecule is then transported into the plant cell. Does that make sense?
Yeah, and it's super cool, actually. I mean, the idea that the signaling exudate kind of like combines with the microbe, and between the two of them, they develop this superpower that it's the only way that what is necessary is produced. I mean, that's pretty cool. That's a very complex complex. Cool. Yeah.
I think it's awesome. I think it's also important to note, though, that what we're talking about there is like a pretty symbiotic relationship. Mm-hmm. And there is going to be some opportunistic stuff too, because, plants are making stuff that maybe it has one purpose, but the microbes can use it for something different. So it uses it for something different and produces some other stuff. And those different types of molecules have like a happy accident. The plant could use some of them too. Like sometimes it is more like a free bin than it is something that's like really organized. But there can be both happening.
All right. I like this idea of invitation microbes too. All right. So if they are all speaking to each other through subtle chemicals, for those of us who are, we're adding things to our soil all the time, this fertilizer or that fertilizer, or, some kind of soil drench with like an FPJ in it or something. How easy is it for us to disrupt that chemical signaling back and forth? Just like, let's say that, I'm talking with a friend in my yard and a plane flies over, relatively low because I live near an airport, and we just have to like stop talking for a minute and wait for the plane to fly over. Is there anything that we could potentially do as cultivators that would be so, air quotes, loud in the rhizosphere that everybody have to stop talking for a minute because we just dumped something in the soil?
Honestly, I think unless it's water, like probably everything we add is disrupting things a little bit. It's a bit like getting a shipment, everybody stops to bring in the delivery kind of thing. I think that is also true of microbes because they're responding to whatever you've added as well. But I think all of them are going to be disruptive. I think it's more how long, because like, for example, if you're just adding water or some nutrients, like depending on how light or how much nutrients you're adding, it might be like really short term and they can use up those nutrients pretty quickly and then they're back to doing whatever. Or maybe it's more long term and you've really altered the soil chemistry enough that like maybe something dies off or those kind of things. I think those are all things that could really disrupt. And I think especially with nutrients, because a lot of what we're talking about is the plant signaling for nutrients or something related to nutrition, that's probably one of the things that would turn off signals from a plant. Like they don't need to get it from the microbe if you're providing it. And I think that might be something else that would really, I don't know, that would definitely be a plane overhead situation.
Sure. I want to take one more step along this disruption idea. Specifically with water, I mean, clearly the soil requires water so that things can have mobility. But sometimes, a cultivator will just overwater terribly and it can, drown lots of varieties of microbes or cause them to cyst up, and depending on the variety. Can we drown exudates too, to the point where, we kind of like wipe out a whole, generation of them, and the soil then needs to dry out, and then the plant needs to restart?
I think yeah, like if you're like watering to runoff and stuff like that, there's certainly stuff that's leaching out of the soil, and some things are kept more by the soil than others. So when we talk about these molecules, a lot of them are charged, and some of the charged, like the positively charged types of molecules tend to stay in soil better than negatively charged ones do. So you could definitely like flush a lot of things out that way. But I think the time scale for production of exudates by the plant, it can replenish pretty quickly. It's not on like the, days, weeks, or months sort of situation. Like those cells are making a lot of stuff really rapidly, and some of the responses can happen within seconds. I don't think it'll be seconds to make the exudates again, but the idea that they might need to make more of them is a pretty quick one. So, I mean, you might disrupt it like really temporarily. I think the biggest danger with overwatering is probably damage to the roots or like promoting the growth of anaerobic bacteria or pathogenic species, that kind of thing.
I follow. So that was, we were just talking about the signaling variety. And so let's talk about the nutrition variety. And it sounds like some of the signaling ones actually do end up playing a role in getting nutrition for the plant themselves as well. What's the difference between like that kind of activity and the ones that we were putting in the nutritive exudates bucket?
I think those ones are more direct. So, I mean, everything is happening at like a chemical level. So the chemistry of the molecules can interact with the chemistry of the soil and just completely bypass the microbes. So, for example, I work with iron. Iron is really hard to get out of the soil. There's lots of it, but it's kind of like it's locked up like a rock. And so the plants will actually make molecules that can go and sort of fish it out from the rock and interact directly with iron molecules. And then that is brought over to the root and it can be taken up that way. Same with things like nitrogen and phosphorus and things like that. I think there's certainly like either they're making it a more favorable environment for those ions to just kind of like those molecules to just be available for uptake, or they're actually able to like drag it in. Some plants, those non-protein amino acids that I was talking about earlier, some plants like grasses will make those and they actually just kind of pick up the iron like a little basket of eggs and actually bring it inside the cell. So they can interact certainly with molecules to signal back and forth to tell them to make other things and bring it in, but they can actually just interact with the nutrients directly as that molecule and bring it in as well. Does that make sense?
It does. So when cultivators kind of like loosely say, you know, oh, this fertilizer is plant ready, or that fertilizer needs to be, you know, processed in the rhizosphere before the plant can take it. Is it these exudates that we're talking about that are doing that work of coming across, you know, nutrition or minerals or whatever in the soil, and the exudates are what are, you know, breaking it down and putting it into a form that can interact with the actual root zone?
That's certainly part of it, yeah, for sure. And like one group we haven't talked about is low molecular weight organic acids, which are like really, really small, basic, what's one that I can think of off the top of my head? Like citric acids and things like that. Plants can make those, and that will just make it easier to take up. Is that sort of what you were talking about?
Yes. Mm-hmm. Yeah.
Yeah.
Because, you know, most of the time we just talk about that, you know, casually using like contemporary language and not the science specific language. So to kind of like, you know, accidentally come across it, it's kind of exciting. It's like, oh, this is what we're talking about all the time and we don't know what we're talking about. That's great.
And there's definitely like this big component of like when we talk about, especially about living soils and things like that, there's everything at the end of the day is a salt, even if it's from an organic material or something like that. It's being broken up into these little tiny ions that we call salts. And the I guess it's just a matter of whether it needs help to get to that state or whether we're providing it as that state. And anytime we're talking about these like exudates helping, like they're basically trying to put it into that form so the plant can take it up.
So then the third bucket we were talking about were, I don't know, for lack of a better term, I'll say allopathic. They're kind of like, I don't know, almost military. They're where they go and they are hurting the plant enemies. Is that like the general idea?
Sort of, yeah. So allelopathic chemicals are these sort of natural herbicides that some, and not all plants make them, but some plants make these types of exudates that basically act as like a herbicide or a fungicide or something like that to clear the way. So it's made by the root's going through the soil and like leaving this material behind that is really toxic to some plants, and that kills off neighboring roots and that kind of thing and lets it grow better. And invasive species are often really good at making these kind of things, and the plants that are here already don't have any defense against them, and so it just kills them off.
So I know I asked you that question in kind of like a general way. Have we found cannabis plants making this particular kind of war-making exudate yet? Have we seen that in cannabis? I mean, I know we're still just learning what cannabis puts out, but have we found that yet?
Not to my knowledge. They're pretty unique situations when this is a thing. Like, for example, walnut trees, black walnut trees, you cannot grow anything under a black walnut tree for a huge radius because that's how toxic it is. But cannabis, you can plant quite a few plants right next to it and it doesn't seem to do anything. So if it is, it would be on maybe like, I don't know, maybe it would be antifungal or something like that. But I don't think it actually has anything like this that would kill off plant roots like that.
That's a great example because that's actually what I was thinking about. I was thinking about, oh, maybe this is why some of the companion planting that we put with cannabis is more successful than others. Maybe the cannabis plant is actually trying to choose its neighbors.
It certainly can. And like all of these root things that we're talking about, like it influences the soil environment, and that environment is going to be hospitable to some other plants and less hospitable to others. Maybe not because it's directly killing off the plants, but just because maybe that plant doesn't enjoy those kind of conditions the same way.
Mm-hmm. All right. So we talked about the signaling kind, and we talked about the allopathic kind, and we talked about the processing that are going to go and free up nutrition in the soil. And then I think the last category that we haven't covered are the exudates that are themselves nutrition. So tell us a little bit about the exudates that the plant puts off that is nutrition itself.
These ones are kind of complicated because I guess it depends on what is necessary, but a lot of things that are carbon based, because carbon is basically what we talk about, like sugars and things like that, are what end up being used to make energy. So I think that a lot of different types of root exudates could be nutritious to other organisms, provided they can break them down and use them. So I think a lot of the metabolites I work with, we think might be getting eaten up by bacteria, which are like the acids and things like that. Anything that has a sugar attached, because a lot of these molecules are combinations of these different types of groups. So you can have an acid that's attached to a sugar, for example. Like all of those would be possible nutrients.
Since you brought up carbon, I want to ask you a question. It's kind of I don't know where this might take us, is what I want to say, is that last night I was talking to soil biologist Leighton Morrison on the phone about how excited I was about our interview today. And he's like, oh, exudates are great. They're like liquid carbon. And I wasn't sure what he meant, but I figured I would ask you about him and say, are exudates like liquid carbon?
A little bit, actually. So a lot of, there's a huge number of atoms out there and elements, but the ones that are most common in organic chemistry and life are carbon, oxygen, nitrogen, phosphorus, hydrogen, and sulfur. And so most of these tiny, it's just those six, and obviously there's other things like iron and plants need manganese and magnesium and all these other different metals. But a lot of these molecules that we're talking about today are based off of those six. So when we're talking liquid carbon, a lot of these molecules have a carbon skeleton, so they're mostly carbon. And as long as something can break the carbon apart and use it in their biology, that's food. Does that make sense?
Mm-hmm, I get it. So I want to talk a little bit about the nature of the signaling. I'm curious to know whether it's more push or pull. When the signaling happens, and perhaps it's both, is it more like putting out a sign that says, I have cookies for sale. If you want cookies, come over to me? Or is it more like placing an order where one side of it, either the microbe or the plant, will put out a request to the other side for what they specifically need?
I think it's sort of both, and it really depends on which specific interaction you're talking about. But I kind of like to think of it more like a conversation. There's going to be some asking questions and I need this, but there's also going to be I'll give you this too. And it also happens over multiple cycles. So it's not just a one message in, one message back sort of thing. These cycles can have multiple signals back and forth. Does that make sense?
Yeah, it does. So where in the plant are the exudates made? Are they made in one particular exudate factory, or is it made all over the plant in many places?
To be honest, I think it's complicated. I think that our general understanding of where exudates are produced is usually the root tip, because that's the growing part of the root, and it's the one that kind of has to be the most responsive to the different things it's encountering as it's moving through the soil. As far as where on the root, it's going to be on the surface cells mostly, because they're the ones that are directly next to the rhizosphere, which makes sense. But I think a lot of different types of molecules and signals to make those molecules can come from all over the plant too. Different parts of the root can ask for different things. Different parts of the shoot, the growing part of the plant that we see all the time, that can also trigger different things to be made as well. But I think the cells right on the outside of the root are the most responsive to that kind of stuff. And it's different too, because above that growing zone, there's still exudates being produced, but they might be different because there's different needs if you're already there.
with all the different needs and all the cacophony and how small this is, I can imagine that there are so many exudates next to each other, that they're nearly tripping over each other because there's so much activity happening.
Mm-hmm. Yeah, a little bit of a soup.
Yeah. So the are the exudates like stuck where they are and they're only mobile if there is water around, or do they have some kind of solution for mobility for themselves?
That kind of depends on the chemistry. So like in general, they're going to be mostly right next to the root, and then they're just going to kind of diffuse to places where there's less of them. Same as like if you were to dump a bunch of salt into the glass, eventually the salt would all dissolve into the water, but they would be most concentrated in the bottom unless you stirred it up. So it's kind of like that if you imagine the bottom of the glass is like the cell. So it's being made right at the bottom there and then kind of diffuses up, and eventually it'll get pretty far away, but not too far. And then it depends on the type of molecule because they're mostly water soluble. But there can be some that are kind of more lipophilic or like oil loving that might move differently. They might be more likely to stay next to the cell that's more like that, and maybe don't travel as far away from the root. Does that make sense?
Yeah, it does. And it doubly makes sense because, the theme of all of your answers have been it's complex and it depends on which one. And I'm starting to realize that, most of the generalizations that we might make about exudates really don't apply to everybody. Like it's hard to make generalizations about exudates. So I guess we'll kind of like wrap up this first set with what is it about exudates that make them be exudates? Because if they all act in different ways and move in different ways and signal in different ways and do different things, there's got to be something that they all have in common that makes them exudates. So what is that?
Honestly, it's just location, I think, is like the biggest thing that connects all of them. Because as you say, there's lots of different functions for them. There's lots of different types. They have lots of different jobs and interact with different things. But they're all outside the root. They're all made by the plant specifically for being out in the rhizosphere. And that's the thing that ends up connecting all of them.
So the exudates are created outside of the root, or it's made in the root and then it's shoved out of the root?
It's made in the root and shoved out. Like I was saying earlier, like those cells that are right on the surface of the root, they're making them inside and then packaging them and then sending them out into the rhizosphere.
That kind of reminds me of the endophytic bacteria that we talked about a couple shows back about, the thing that makes it a bacteria endophytic is that it spends some amount of its life inside of the plant. And so it goes, it'll go in and out of the plant. And so perhaps exudates are, things that start in the roots and then they are pushed out. Does that make sense? I don't know. I'm making it up.
100%. All right. Yep. Nope. That works for me. That's as good a definition as any.
All right. Great. Okay. Let's go ahead and take a short break and be right back. You are listening to Shaping Fire, and my guest today is plant biologist Sarah Lane. And, without these advertisers, Shaping Fire would not happen. So please support them and let them know you heard them on Shaping Fire. Online cannabis seed distributors often seem to be all the same, but Multiverse Beans constantly works to provide you with cannabis seeds and a buying experience that you won't find elsewhere. Multiverse Beans works directly with the breeders to secure as many packs of your favorites as possible so that they have your favorite beans long after others have sold out. Some shops simply buy breeder minimums, but I get messages all the time from breeders saying some version of Multiverse asked to buy my entire run. At multiversebeans.com, you can find rare cannabis seeds from Night Owl Seeds, including the Dark Owl sublabel, Mephisto Genetics, Square One Genetics, Robin Hood Seeds, and Ethos, and so many others. Multiverse also initiates projects with breeders to secure exclusive packs that you simply won't find elsewhere. Multiverse founder Paul Lal sees himself not only as a curator of the best cannabis seeds available, but also as a collaborator with breeders, trying to bring novel crosses to the market that his customers are asking for. Multiverse Beans also creates exclusive stickers for their popular seed varieties that are available free only when you order those seeds from Multiverse. Check out their stickers like the badass recent slap for Mothman by Gnome Automatics on Instagram at Multiverse Beans. And finally, the freebies. As you'd expect, Paul sends quality freebies with every order. And when you spend at least $150, Multiverse allows you to choose your freebies from their special selections. You can get a 10% discount off regularly priced items when you use the discount code SHAPINGFIRE, all one word, at checkout. Sign up for their mailing list to be eligible for their monthly seed giveaway worth $250. So go to multiversebeans.com now for a buying experience you won't get anywhere else. For years, organic cultivators have been looking for a peat moss replacement. Peat moss has long been the go-to soil amendment for water retention and container growing, but organic growers are recognizing now that peat moss is an unsustainable resource, and the mining of peat bogs destroys wetland habitats and releases sequestered carbon. But peat moss works so well that many have continued to use it. Now there is finally a revolutionary replacement for peat moss that provides better benefits while being a sustainable choice. Pit moss sounds and acts like peat moss, but instead of being mined from fragile ecosystems, is actually made from upcycled organic paper and cardboard headed for landfills. Pit moss is excellent at retaining water in your substrate and creating air pockets and tiny living environments for microbes. Pit moss instantly increases aeration, nutrient absorption, and water conservation too. Carefully and locally sourced, pit moss is the result of decades-long research into the use of recycled paper fibers. Pit moss is lightweight and easy to use, and pit moss is inert, so it won't change your pH. Available in a range of preparations, including a nutrient-enhanced blend and an organic soil conditioner with no added nutrients. Pit moss is also available as an animal bedding for horses, chickens, and small animals. You can save 15% with the discount code SHAPINGFIRE, one word, no caps, when shopping on pitmoss.com. So go to pitmoss.com now to learn more. That's PITTMOSS.com. Growing healthier, stronger, more sustainable plants. Pit moss. One of the reasons why no-till cannabis growing is so valued by farmers is because the mycelium networks in the soil remain established from year to year. And we know these fungal networks are essential because they are the nutrient superhighways that extend far and wide in the substrate to feed your plants. The trouble with growing in new living soils or blended cocoa substrates is that it takes most of the plant's life just to create these mycelium highways. Dynamyco endomycorrhizal fungi inoculant reduces that time and gets your plant eating a wider array of nutrients faster. And it's 3 times the concentration of the other popular brand in the US at 900 propagules per gram of 2 fungal species selected specifically for cannabis cultivation. Dynamyco is the result of 30 years of research and trials at the Volcani Agriculture Research Institute in Israel. It has also been vigorously trialed by cannabis and food growers across the U.S. Dynamyco is now available at grow shops and on Amazon in the United States. I love using Dynamyco to both speed up the growth of the mycelium networks in the soil, but also as a biostimulant to make clone cuttings more virile. You can see side-by-sides showing the comparative growth on their Instagram at Dynamyco. If you demand reliable growing results and appreciate the importance of an active root zone in creating a thriving plant, I encourage you to check out dynamyco.com and use the store locator to find out where you can get yours. That's dynomyco.com. Shaping Fire listeners can get 10% off any size of Dynamyco on Amazon or dynamyco.com by using the discount code SHAPINGFIRE, one word, no caps. Whether you are starting with new beds or pots, or if you want to add some zing to tired soil, choose Dynamyco to maximize your plant's potential. Dynamyco endomycorrhizal inoculant. Welcome back. You are listening to Shaping Fire. I am your host, Shango Los, and my guest today is plant biologist Sarah Lane. So during the first set, we talked a lot about kind of exudate mechanics and what they are, so that we could get an idea of get our heads wrapped around this interesting chemical that's in the rhizosphere and what it does. And so on during this set, we're going to focus more on what they actually do and how this, I don't know, cacophony or symphony takes place. So how specific are the signals for either nutrition or the signals to telling exudates to go out and mine some manganese or something? Are there specific exudates for all of these processes, or is it like when you tell somebody, hey, would you pick up bread, but I don't tell them what kind of bread? I'm trying to get an idea of how specific the requests are.
Ooh, another theme of the day. It's complicated. I think that in some cases it's extremely specific, especially when we're talking about symbiotic relationships like rhizobium, or in legumes or mycorrhizae and that kind of thing. It's very specific, to the point where it's species specific. It's a specific molecule that has one job, and that is to go and signal to mycorrhizae fungal interactions and things like that. So it can be that specific, but there's also some that have sort of more of a broad, it has multiple different types of chemistry. It might be maybe a more complicated molecule where one end does one thing and the other end does another thing. And sometimes the plant can make multiple things to do the same job.
So some of the tasks that the exudates do, especially the ones that are sent out for nutrition, and probably similarly the ones that are sent out for like inert things like manganese or iron or whatever, those jobs sound familiar or similar to the role of mycorrhiza. And we often talk on this show about how the fungal networks are like super highways that will go out and source these different nutritions that are needed by the plant, and then kind of bring them back. And there's this relationship there. I would think that since this task is very similar to the exudates, or at least how they're described, there's probably a lot of exudate interaction with the fungal network. Is this true?
Yeah, I would say that's true. So I think there's two components. One, the exudates can do a similar job, but on a much more tiny scale, because they don't travel very far. And so if they need to go farther out for the nutrients, then the exudates would interact with the mycorrhizae directly. And a lot of that is basically initiating infection. It sounds weird to say infection when I don't mean pathogenic, but essentially the plant has to invite the mycorrhizae, especially endomycorrhizae, to infect it. And that's a very specific signal that's sent out to interact with that fungus to allow it to come and join the plant. And it has to be in a situation where the plant would need that. So I think if the plant is deficient in phosphorus, for example, it might interact with mycorrhizae more. Whereas if it's not that deficient, or if there's plenty of phosphorus next to the root, maybe it interacts with that less, and the exudates that it makes for itself will do the job. Does that make sense?
Yeah, that does make sense. Is it possible to overtax the exudate system? So if the plant is, making these exudates, and everything that the plant does takes some amount of, I don't know, energy or life force or effort, depending on what level we're looking at. If the exudates are so busy, can the exudates be working so hard that it makes the plant less thriving?
I think that kind of depends. So I think if the plant is unhealthy, that might be true. We often talk about things like a carbon budget, though. So there's a certain amount of carbon that a given amount of plant can fix, and there's a given amount of nutrients it can take up just based on its biology and physiology. And so carbon budgeting that means that different parts of the plant will be allocating carbon to different places. So if, for example, it's suffering a massive thrip wave, it will be busy making carbon containing compounds to deal with that, and it might make less exudates. But if it's perfectly healthy, all things considered, it will make what it needs, and it'll make the amount that it needs. Does that make sense?
Yeah, it does. So So what I was going to ask was that, are there things that we can do that would stress the exudate process and signaling exchange? And then you threw the thrip thing on the end. So perhaps the answer to the question that I was going to ask is, like, anything that stresses the plant is going to call on the exudates to have to do more work. So don't stress your plant.
Basically, yeah. Or it's going to divert the resources of the plant elsewhere. I think, like, there's a certain amount, we call it basal or base. There's a certain amount of, like, maintenance level things that the plant makes everywhere. But it will respond to different stresses by taking as much carbon and nutrients as it can to whatever that is. So if it's happy and healthy, then it can, like, pump up those base levels and everything's great. If it's really under stress somewhere else, it might take what it would normally make in the roots. It might take some carbon from that and send it somewhere else. And that might mean that there's still probably going to be some exudates there, but it'll probably mean that there's less of them. Does that make sense?
Yeah, it does. And so for somebody who's listening to what you're saying, like me, and my brain goes to, what can I do to not stress out the exudate creation? Like, that's nice that I'm thinking about that level of my plant, but really that's not really necessary. I really should go up a level of magnitude and think about not doing things that stress out the plant, and then just let the exudates manage themselves.
Essentially, yeah. Like, I think if you take care of your soil and you take care of your plant and they're both in good shape, then you shouldn't have to worry about it for sure. Okay. I mean, making sure that your soil is healthy is definitely a big part of it. Making sure the plant isn't, being, as I said, mauled by thrips or something like that is also helpful. I don't think anybody wants that anyway, but those kind of things, making sure it's getting enough water, it's not overwatered, all those things that we generally try and make sure our plants have anyway. If your plant is healthy, there's not really anything extra that you need to do. I think I think compost teas are one thing that you can do to kind of help with soil life and things like that. And that might help with the exudate situation, but most of the time it's really out of your control.
Well, it's kind of nice to have something that, we're learning about this process, but it isn't another thing that we have to, like, technically worry about, right? If you do your regular living soil kind of stuff, like this will take care of itself, which is great because we already have enough to worry about, right?
Yep.
We'll talk more about compost tea a little bit later, but I want to wade into a pretty fierce debate in the cannabis world, and I'm a pretty heavy participant in it, and I have been looking for someone like you to weigh in on it. So I'm gonna pull you into this debate, and this is on 24-hour light cycle. So okay. The debate is generally about does the 24-hour light cycle, is it best for the plant, or does it starve the soil from nutrients? And the idea kind of goes like this. Some growers like to use 24 hours light because it causes the plant to grow more quickly, and you can either grow a bigger, higher yielding plant during that time, or during its regular life cycle, because it's just getting more light, or you can actually shorten the cycle by using more light, which allows you to perhaps get in another growing cycle over the year. And so a lot of people use 24 hours of light. Now, the flip of it, which happens to be where I stand, is that no, 24 hours of light indoors is not going to create the best plant, because while it makes it grow faster, you're not allowing the back and forth of the light phase and the dark phase. Both phases are necessary so that the exudates and the signaling and all the processes that happen when the plant is awake with the light it needs to have some dark times so the contrasting signaling and exudates can happen. So you need both, so all varieties of exudate signaling can happen, and all of the varieties of nutrition and those things can go back and forth. So I have a feeling that the answer is probably somewhere in the middle. But specifically to this 24 hours of light question, how does 24 hours of light impact this relationship between the plant and the rhizosphere as far as the exudates playing a role?
Oof, that is a spicy debate, isn't it?
Yeah.
I think, again, to the theme of the day, it's complicated. I have not ever seen anybody be able to, because of the complexity of the rhizosphere and all the stuff that's going on there, I haven't seen anybody actually be able to put this into a scientific study where we're able to say definitively, if you give a plant 24 hours of light, the exudates suffer. So it's really in the middle, I think. There's a couple different things that I can think of off the top of my head, if it's okay if I ramble for a second.
No, please. We want to get into this, so go ahead.
Perfect. Okay, so light cycles. Light cycles, I think lots of things are turned on by the plant rather than off when light is on. And that's because light is driving water transport and nutrient uptake, and it's also driving photosynthesis. So I think that too much dark is as bad as too much light is, if that makes sense. Like, you're seeing the growth and the yield increase because the plant is doing a lot of stuff during that time. And there is a circadian clock in plants where there are different things that happen in the daytime and in the nighttime. And they actually are different for the root part and the shoot part. So they actually operate on different clocks. But they do talk to each other. So I guess where I'm going with this is that there is a circadian rhythm, but we don't know enough about it to be able to say that having light on the top of the plant is going to affect the roots in the way that I think this debate kind of goes. Mm-hmm. Because if we think about it, the roots are always in the dark. They're never in the light. They're not, like, there might be a small amount of light that makes it to the root, but not enough that it should matter. But they can still sense it a little bit. Man, that is a very deep topic.
Yeah, it is. And so maybe I'm thinking too top down about this, because even though the light doesn't specifically reach the roots, right? During the light mode, the plant, I think of it as the plant is giving commands through the roots. And so even though the roots aren't lit up, the roots are responding to needs and commands from a plant that is actively photosynthesizing. And then in the dark cycle, those commands and needs of the photosynthesis are no longer taking place. And so I imagine that the roles and the tasks of the root zone change when the plant is changing modes from photosynthesizing to, the processing of nutrients that it does during the dark.
Mm-hmm, exactly. I think the key word there is change, because if they take up a bunch of nutrients during the daytime, then a lot of those exudates that are helping with nutrient acquisition should be active. Like, they're actively, fishing out the different components that it needs and putting it into the plant so the plant can continue to grow. And those would change if the light is gone. And they do have, we even know from those green potatoes, roots themselves can sense light, both outside sources of light, but also the inside sources that we were just talking about, the light that the plant above is sensing is also they notify the roots that they need stuff, right?
I think this next question is the seal the deal question that I've been looking for. So if the root zone is doing different tasks when the plant is experiencing light, because the plant is asking for things involved with, photosynthesis, production during the day, those types of tasks, and then when the light is gone, the root zone changes to different sets of tasks. that would seem to suggest to me that it needs a dark period so those dark period tasks can happen. So maybe I'm right, but for the wrong reason, for a reason I didn't realize. I thought that during the dark, the exudates would stop. And maybe that's not it, because exudates happen around the clock. Maybe it's that the exudate and signaling that happens when the plant above is experiencing dark, those differentiated tasks need to have an opportunity to happen to have a thriving plant. Yeah?
Yeah, I think that could be a reasonable way to go about it. I think that there's also two things. I guess there is dark reactions, and they're like evening onset types of things that change in the plant that are responding to a lessening of light, at least when we're not talking about like growing light, when we have actually like changes in light over the daytime as well. Your plant on the balcony or whatever. But there's also light independent reactions that tend to happen kind of at any time. And what I mean by that is like photosynthesis is light driven. Like you absolutely have to have light for that whole series of chemistry reactions to happen. Whereas the like other types of reactions don't need light like that, so they might be happening 24/7. What I really think, for my own personal opinion, definitely an opinion, is that a little bit of darkness helps, because one thing that light is a stressor. It creates all these like fast happening reactions. It's quite powerful. It can make really reactive molecules that if they're present in too much, they can damage the plant, and those kind of things. So I think having 24 hours of light constantly would, maybe mean that you don't have the downtime that you need to take care of any kind of damage that's coming from light and that kind of stuff. So for, I guess, my way in on this would be like a little bit of dark is helpful, I think. Does that make sense?
Yeah, it does. And I'm grateful to hear it. So this idea that there can be too much light is interesting, because, most of us or folks who grow indoors anyway, while I've been historically an outdoor grower, I've been dabbling in indoor just to learn new stuff. And most of the time, cultivators are looking to blast the plants with as much light as is possible without bleaching or burning the plant. And what I'm hearing from you is that there really is a top end of how much light we necessarily want to give the plant, because there is too much. There's a top level.
Yes, there is. I think especially with like I have most of my experiences is growing inside. And I think there's different things to think about, because you can definitely have too much light inside, but it's not like an overall amount of light. There's all these focal lengths that come with bulbs, and it's like shining a laser, the magnifying glass on the, something flammable in the sun kind of thing. Like it focuses all the light from the bulb to a single point. And if you're growing in that point, it's really strong, but like just for that little area. And that's too much light. But there's so much sun out there. I don't think any indoor bulb at this point has ever gotten to the point where it can compete with the sun. It's just too dim. The spectrum isn't strong enough. And a lot of the damaging wavelengths that we think of with light, like UV B or C, are just not present in the same quantity unless it's like, a UVC light or something like that. Does that make sense?
Yeah.
And so there's too much point light, and then there's too much general light.
Yeah, I was making an overgeneralization about the too much light as if it was all sunlight, but that's true. Like all, no matter what kind of bulb we are talking about, it is some degree less than sunshine. And so that's probably that gap between sunshine and whatever the personality of the bulb is. That's probably where the hitch and the giddy up is, right? Is somewhere in what the bulb is lacking when compared to the sun.
Mm-hmm. Yeah. So I think indoors, like most of the light stress that I've seen is like, you turn the lights on right after you spray something on the plants, and you get some like lens burning where the light's just kind of magnified and focused at a point. So you get like little point burns. Outside, you can definitely see a lot more types of light damage and stress because it's so bright out. And if it's hot too, like the plant just has a really hard time managing that level of light in that those poor little photosystems get a little stressed.
Mm-hmm. when we talked on the phone the other day, kind of preparing for this discussion, you used a term that I really liked, and I don't know if it's a scientific term or if it's just something that you were using to describe to me, because you use such fantastic examples. But you use this phrase, free living microbes, and I remember talking to you. I remember like, oh wow, I never really thought about microbes that were in the soil but weren't really interacting with the plant. Would you just talk about free living microbes for a moment?
Absolutely. So essentially, when we think about ecology and like what's happening in the soil, we definitely talk a lot about microbes that interact with the plant, but there's a couple different levels they can do that. There's symbiosis, where they need each other, and then there's kind of like opportunistic sort of interactions where they don't. So when I talk about free living microbes, we generally use that term to mean things that are just, they're in the soil too. They don't have to be next to a plant. They don't, like if you remove the plant, they go on living, they do their thing, and they it's not the same. And if anything, like maybe the plant needs them more than they need it, if that makes sense. Mm-hmm. But if they're free living, they can complete their entire life cycle without a plant, if that makes sense.
Mm-hmm. So that also brings up an interesting question about the opposite. So you said, oh, there are some of these microbes that wouldn't exist without the plant. Is this one of the reasons why it is so helpful to the cannabis plant to have other companion plants around, because these other plants are helping support these free living microbes, and it just makes the entire rhizosphere more complex and more thriving and more abundant?
Oh, I'm sure that's true. Yeah, because like cover plants, for example, essentially what they're trying to do is improve the soil life, and that happens either through like providing a matrix, like all those roots provide basically housing in a lot of ways, like a little scaffold for different things to live on and in and under. But it also might help with, for example, maintaining humidity longer, so that if there's microbes in the soil that prefer to be wet all the time and they don't like drying out, then you might have that layer is happier because there's a plant there keeping the moisture locked in. Maybe they don't like a lot of light and they're kind of shading the surface of the soil a little bit, so maybe the fungus can get a little bit more close to the surface or something like that. Like I'm sure there's dozens of different things that a companion plant. And we also plant, like I personally plant clover, because it tends to also, it has an interesting relationship with bacteria that actually lets it take on and fix more nitrogen than a lot of other plants do. But it also brings the benefit of having that nitrogen fixing relationship with these bacteria that also benefit plants around it. So the cannabis plant itself doesn't do this, but the fact that there's a plant near it that does improves the nutrients in the soil. Mm-hmm.
One of my favorite reasons, and the first reason I started companion planting for my cannabis, is I had earlier in my cultivation history, I had a tendency to water incorrectly. I was doing too much, not often enough. So I needed to water more often, but less. And I had a tendency to get hydrophobic soil. And for folks who aren't familiar with that, that means that the top of your soil container gets kind of hard. And then when you go and you hand water, the water just kind of slides off and goes down the side instead of actually soaking into the soil. And this happened to me all the time. And I eventually learned during the Shaping Fire Water and Watering episode that if you use a companion plant, or in this case, what we would consider a cover crop that has lots of roots, say for example a clover or some types of grasses, they not only can provide like the canopy, if you will, to create this little moisture vapor area between the tops of its leaves and the soil, so the soil just generally stays more moist. But also, I was going for the fact that all of these roots that go down into the soil all are providing like small tunnels for the water to actually go into the soil itself. And, I actually solved my poor watering just by adding a multiple root plant, and then that problem just went away.
Yep, that's and the other wonderful thing that you're doing is you're also introducing different types of exudates, which improves your soil life. So then you're just getting, it's just more all the way around.
So are we aware of any, either companion plants in cannabis? Of course, we use the idea of companion plants to often be the idea of like a bigger plant that's we're planting nearby, like a cabbage or potatoes underneath it or whatever. And then we kind of use the term cover crop, even though most of us in cannabis use that term not how it's traditionally used, to talk about lower plants that there's a lot of them. I'm kind of backing into this question, but what I want to ask you is, are there plants that you are aware of that share exudate flavors, complementary exudate flavors with cannabis plants that we should grow around our cannabis plants simply because they increase the vibrancy of the kinds of nutrition our cannabis plant is going to want, thus suggesting, oh, you should grow these plants because it too will attract the kinds of nutrition and make it more buffet style for your cannabis plant?
Ooh, I think that the honest answer to that one is I don't think we're anywhere near that level of detail in the exudate scientific realm. Like, even just to identify exudates is super challenging because you can't even grow them in soil to be able to collect them. So all of my experiments, for example, are done in a fogponic setup that I made, so that I can actually access the roots without the soil, because of course otherwise you capture all the exudates from all the bacteria too. So I don't think that we could get that specific, but I do know that they've been doing a lot of interesting research with crops and companion plants and crops, and the running theme there is usually something that fixes nitrogen. Really helps because it brings in not only the nitrogen fixing aspect, which is usually the limiting factor in plant growth, but it also brings all of its symbiotic bacteria with it. But I think as long as you have a cover crop, as we were just talking about, that's improving your soil texture or soil health in general, I think that you're good. And so for me, like the anecdotal answer would be something like we plant clover, partly for the nitrogen fixing, but we also plant things like basil or marigolds that can act like trap plants to kind of give us a sense of whether or not we have pest things to worry about, and that kind of thing, or anything that helps like pollinators come in and that kind of thing, because that usually, I don't know, more life is better usually. Does that make sense?
Yeah, I follow that. The example that you gave, I had not really considered how difficult your research must be, because I figured there was just some way for you to, grab the exudates, scrape them off, pull up a plant and scrape some off the roots, that were in soil. But that's right. There's everybody else who's in there too, especially the bacteria. They're all laying out exudates too, and so it's just like a mess, and you can't tease them out. And so you're using this fogponic system, so the roots are just dangling, and there's not soil life interacting with it. So in an odd way, you're like an exudate rancher. Like you've got to do this fogponics so that you can collect them first.
Yep, that's correct. Yeah, we have a couple other ways, but they're all like they're all complicated. You can do sterile culture, like tissue culture. But then the necessity of that is you have to have a plant that you can grow in tissue culture. And if I was working with cannabis, I think it would be easier because it does grow in tissue culture, which would be nice. And then you can just sample the media and extract the exudates from the media. But there's lots and lots of plants out there that don't like tissue culture, that won't grow in a little tiny box, or trees, some of which I'm working with right now, that just defy hydroponics in general. So yeah, you got to get clever about it for sure. And it is very much like ranching.
Right on. All right, cool. So let's go ahead and take our second break. During the third set, stick with us because we're going to be talking about salt-based nutrients and exudates, hydroponics and exudates, and unsulfured molasses in exudates. So come on back. You are listening to Shaping Fire, and my guest today is plant biologist Sarah Lane. As cannabis regulations become more demanding and consumers become more educated, it is increasingly important to avoid the use of chemical pesticides when cultivating cannabis. Beneficial insects have been used for decades by the greenhouse vegetable and ornamental plant industry, and today many cannabis cultivators are moving from sprays and chemicals to beneficial insects. Copert has the beneficial insects, mites and nematodes, microbials, sticky cards, and air distribution units you need to partner with nature to defend your garden. Whether you manage acres of canopy or have a simple grow tent in your home, Copert is ready to help answer your questions and help you transition away from chemical sprays towards clean and natural solutions. Since 1967, Copert has assisted growers in identifying pests and devising reliable solutions while providing healthy insects and mites that will protect your yield. Since the 1990s, Copert has been a leader in cannabis pest and disease control worldwide and have highly trained consultants to assist you in Canada and the United States from coast to coast. With their global network of grower support, Copert can help. Visit Copert.com, choose your country, and get detailed information. That's Copert, Koppert.com. For the most up-to-date cannabis-related biological control information, you can also check their Instagram at Copert Canada. You know getting away from pesticides is good for health and good for business, and Copert is ready to help. Visit Koppert.com today. Once you've discovered the benefits of using cannabis, it's a very small step to start making your own edibles, gummies, lotions, tinctures, and concentrated oils at home. Magical Butter has been helping cannabis consumers become self-sufficient for over a decade. With the easy-to-use Magical Butter countertop botanical extractor, you can create high-quality cannabis products to your exact specifications at a fraction of the cost of store-bought edibles. I talk a lot on this show about the importance of home growing so you don't have to rely on others to feel healthy. Well, the Magical Butter machine can empower your personal health by putting you in control of how you use cannabis in your daily life. I've been making my own butters and oils on the stove for years, and I much prefer the ease of using the Magical Butter machine. I just set it and walk away. With the simple touch of a button, the Magical Butter machine grinds, heats, stirs, and steeps your herbal extract, all at the correct time interval and temperature for the perfect infusion every time. As a result, you achieve your desired infusion easily, safely, and consistently. Check out the Magical Butter Instagram to see the machine in action. And don't feel like you have to go it alone. There is a huge community on Facebook called Magical Butter Users United, sharing recipes and best practices so you can learn at your own pace from others who are already doing it successfully. Now is the time to get your own Magical Butter machine and save money while enjoying cannabis. Use the discount code SHAPINGFIRE, one word, no caps, to get 10% off. Visit magicalbutter.com today. After you've caught up on the latest Shaping Fire episodes, do you sometimes wish there was more cannabis education available to learn? Well, we got you. Shaping Fire has a fabulous YouTube channel with content not found on the podcast. When I attend conventions to speak or moderate panels, I always record them and bring the content home for you to watch. The Shango Los YouTube channel has world-class speakers including Zoe Sigmund's lecture, Understanding Your Endocannabinoid System, Kevin Jodrey of Wonderland Nursery talking about breeding cannabis for the best terpene profile, Frenchie Cannoli's Lost Art of the Hashishin presentation, Nicholas Mahmood on regenerative and polyculture cannabis growing, Dr. Sunil Agrawal on the history of cannabis medicine around the world, Eric Vlosky and Josh Rutherford on solventless extraction, and Jeff Lowenfels on the soil food web. There are several presentations from Dr. Ethan Russo on terpenes and the endocannabinoid system too. While there, be sure to check out the 3 10-part Shaping Fire Sessions series, one with Kevin Jodrey, one with Dr. Ethan Russo, and one with Jeff Lowenfels. And even my own presentations on how to approach finding your dream job in cannabis and why we choose cannabis business even though the risks are so high. As of today, there's over 200 videos that you can check out for free. So go to youtube.com/Shango Los or click on the link in the newsletter. Welcome back. You are listening to Shaping Fire, and I am your host, Shango Los, and my guest today is plant biologist Sarah Lane. So let's talk about a couple of the applications of exudates with common situations that we deal with as cannabis cultivators. And the first one I want to talk about with you, Sarah, is salt-based nutrients. So, on this show, we very specifically support living soil, a biomimicry of the natural world, and are not big fans of salt-based nutrients generally. But, I always know I need to give them a fair shake when it comes to science, because that's really what we're listening to, is the science, not my own biases. So I'm curious to know what kind of a relationship that using, bottled salt-based nutrients has on the exudates and their ability to communicate and the community, I guess.
I think that's, I think most of the time when we're talking salt-based nutrients, it doesn't really go very well in living soil, which is my preferred method of growing too. So we're on the same page there, I think. With salt-based nutrients, there's sort of two things that happen. One is, like we were talking about earlier, it can really disrupt the signaling, or at least that's my theory, because you're introducing a huge amount of nutrients to the plant that it would have been making all of these wonderful little exudates to try and tease out from, as we were saying earlier, microbes or the soil itself, and you're just flooding it. So I think that it would pretty significantly disrupt the exudate type, at least, of what's being produced. The other thing it does is it really kind of messes around with the chemistry of the soil. So if you're only feeding salt-based nutrients, you end up with salt buildup and stuff like that. And that can happen in a living soil system too. And usually those conditions aren't really conducive to microbial life or root health, and you end up just getting a lot of compounding problems.
So it's less about the salt-based nutrients somehow masking the chemical signatures or anything. That it's just that the people who are normally giving off the exudates and receiving the exudates, they're generally just not there.
Yeah, I think the plant, if it's got all the nutrients it needs, it's going to put its resources elsewhere, and I think if you're compromising the soil life at all, then a lot of that signaling would be, as we were saying earlier, probably dampened would be my guess.
Dampened, that's a good word.
Yeah.
A lot of people are playing with soilless mediums that are increasingly more and more, cocoa-based, cocoa coir, to the point that some folks are just like they don't have soil in their substrate, right? It's not really hydroponic. It's just it's cocoa and its nutrients. And I'm curious if these types of cocoa and soilless mediums have any impact on the signaling capacity of these exudates. I guess what I'm asking is the soil component key to the ability of the exudates to be able to do their job?
That's actually a really good question, and I was just thinking about that because of what your previous question was. There are circumstances, so I think the real problem I have with salt-based nutrients is if it's in combination with living soil, because in living soil, you're relying on this microbial life to help provide nutrients, and it just gets all out of whack. But for example, in my research, I have to use salt-based nutrients because I need a very defined set of nutrients that to be able to repeat my experiments. And in that case, with the fogponics and that kind of thing, it's not necessarily like the exudates are going away. It's just that the types of exudates that I'm looking at are nutrient-based and not microbial-based, so I don't notice a difference. And in that case, I think in cocoa or peat or something like that, as long as you're not relying on the bacteria that or fungus that are present to assist you in the nutrients, it wouldn't make as much of a difference. I think it would change the expression of the exudates, though. The types of exudates that are produced would be different. Does that make sense?
Yeah.
Because you're still going to be producing them.
And it's really interesting for you as a living soil cultivator at home, but needing to use salts in the lab. It puts you in a unique position to be, comparing and contrasting them. Let's talk about if you're using salt fertilizers in a soilless medium like cocoa. And you just said that if you were in that kind of environment, it wouldn't that the exudates wouldn't be there. It would be that there would be nutrition-based exudates. You would just be changing the variety of exudates that are there that are being expressed. My question would be, would that impact how the plant expresses itself? It seemed to me that if we are changing the exudates that are part of the cacophony in the soil, that we're going to get, different colors on the plant, different terpene profile, maybe a difference in quality of thriving. It seems like the plant would express itself at the top level. Is there any weight to that?
Yeah, I think so. I think, like, if we were to go back to our bazaar metaphor from earlier, when you feed salt-based nutrients, it's kind of like putting in a really big box store or something like that. It really changes the flavor of what's happening in that location. And I think that changes the personality of the soil. And I think, like, to borrow personifying my soil now, but, the conversations that are happening and the types of players that are there changes pretty significantly. And I don't doubt that would also change the plant itself. The plant is very flexible. It's going to do its best to grow and to thrive in whatever circumstances that you put it in. But when you change those circumstances, it's going to change itself to fit. And so, and I know lots of people that anecdotally say that terpenes are different and like cannabinoid profiles are different when you grow in living soil and stuff like that. Whereas like when you're feeding salt-based, maybe you're going for weight and the plant is forced into putting more weight on because it's got all these extra nutrients and maybe it makes less. And I don't know that for sure because I haven't studied it myself, but I think that anecdotally I've seen that happen.
Mm-hmm, right on. So even though you haven't seen the science for it, it makes intuitive sense.
Mm-hmm. Yeah, I would say so.
Right
yeah.
on.
And like one of the reasons I like living soil is because it sort of gives the plant its own options. Like it's complex. It's kind of a fun way to think about ecology too in a pot, but the plant is really able to pick and choose a little better than kind of, force feeding it salt, if that makes sense.
Yeah, for sure. And we talk a lot on Shaping Fire about the importance of, a complex and thriving rhizosphere to have a complex and thriving plant so that you have a complex and delightful terpene profile. And the more you're simplifying what is in the pot, say, for example, using a cocoa soilless medium and salt-based nutrients, I think the example you used was just putting them in a big box store. So you're going to get big box terps out of it and not necessarily a full range, perhaps.
Yeah, I think that's totally reasonable. And again, like this also ties into carbon budget because when you're forcing a plant to grow in a specific way, you're also asking it to allocate all its resources in that direction too, which generally means it has less for other stuff, which maybe is stuff that we want, like terpenes.
Right on. Let's talk a little bit about. I think I can probably group hydroponics and fogponics together, but if not, tease them back apart for me. I'm curious because we've talked significantly on this show about the soil food web, and then somewhat about the water food web, and a hydroponics solution and I'm assuming a fog solution. We're talking about the water food web, and I'm wondering if the quality and number and variety of the exudates that are in those systems sound like there's going to be less, since you initiated this by saying, I use a fogponics because we're trying to ranch these particular exudates, and in soil there's just too many. So I'm assuming that in hydroponics and in fogponics that there are going to be less exudates based on what you had said earlier. And so with hydroponics, would this same idea cross over from the soilless mediums, that there may be reasons you want to use hydroponics, but in the end there are going to be less varieties of exudates, and signaling conversation going on, and nutrition. So you would run into the same problem with hydroponic systems, that you're not aggressively supplementing,
I totally
I guess.
So.
Yeah.
Yeah, I think that's fair. I mean, for me, the difference between fogponics and hydroponics, just quickly, would be just like I don't know, dryness, if that makes sense. Because the fogponics is, even though it's nutrients, it's very dry. Like the roots aren't sitting in water. They're not like soggy. So anything that's really intolerant of flooding, I've been able to grow more successfully in fogponics, as opposed to hydroponics, where a lot of hydroponics, your plants are really sitting in water or constantly sprayed by water or a lot of stuff like that. But it's a really subtle difference, I think, because the nutrient solution I use for both is the same. So as far as like, it compares pretty well to soilless. I think the biggest difference, at least for me, wouldn't necessarily be in exudates. It would be more in like root architecture, because the plants in a like peat or cocoa, it's more like soil for like how they can grow. But on the exudate level, I think that the same is probably true. There's going to be a more defined set of exudates. They're probably coming mostly from the plant, and you miss out on some of those interactions with bacteria and fungus and stuff like that. And there's still maybe some that can grow in those kind of conditions, but a lot less than would be growing in soil, at least as far as I can tell.
Right on. I also want to add a little something for all my hydroponics people who are listening, who are now like pissed at me because I'm underselling their craft. I want to be clear that I do understand that some hydroponics, there's a lot of varieties of hydroponics. I mean, we did a really great soil food web show couple years ago with Steve Reisner, and he does the first of all, he is heavy into bringing in water with mulm and fish poop in it, so there's a lot of complexity there, and he was describing the great flowers that he pulls from his setup, but he also has some soil in the mix, right? So he's like flooding and draining the soil, and he's got the fish poop, and so he's making this like incredibly biologically active environment that makes flowers with terpene profiles that he says we don't even get in a straight soil medium. So I want to be clear, when I'm talking about the limitations of hydroponics, I'm talking about the kinds of stuff that people are generally either doing in the extra room in their house, or at wild scale of corporate cannabis. I absolutely appreciate the fact that there are really tasty ways to do complex hydroponics. So
that totally makes sense. And I would like to add there that like each of these growing methods has its place, and the plant will grow regardless. And so I know I've had some really good hydroponic weed before. I know that I've had some really good living soil weed. Like they all have their way of doing things. And I also think that there's ways to make hydroponics more accessible to bacterial life. And like you were just saying, with a different terpene profile that you don't get with either of the other two. And that just kind of goes to prove the point that what you're putting on your roots does change. And same with the type of life that you're growing changes what the plant is able to do. And sometimes that's for the better, depending on which method you're using. So definitely, like, whatever you're growing your plant in is better than not growing your plant at all.
Yeah, there you go. And, that's one of the challenges that we ran into during these, like, 24 hours of lights debate. People are like, I grew a huge ass plant that was absolutely beautiful and totally got me high, and all my friends bought it from me, and that was great. And they're all like, why are you telling me that I shouldn't be using 24 hours? And my response is always, it's not that I'm saying that you can't grow plants and that your plants are going to suck if you go 24 hours. I'm saying that you're probably leaving some quality on the table by doing it 24 hours, and your terpene profile could be better, and the plants could be better, and you could be more pest resistant if you just gave it 4 hours of sleep or 6 hours of sleep. It would be helpful. But internet debates are rarely that nuanced,
Oh, it's so true. And I do think that, like, even it doesn't really at the end of the day matter if you're growing a plant that suits your tastes. Like, however you're doing it is great. I mean, unless you're dumping like illegal pesticides on it or something like that, maybe don't do that. But, like, I think at the end of the day, everybody's going to have a way of growing that works for them. And there's absolutely, these debates are all like trying to tease out the finer ways of growing and like the best way to grow. And that's going to be very much personal preference at the end of the day. And I personally prefer the taste of living soil weed, but I've had really good weed that's been grown in every other condition you can imagine. So I'm, here. As long as you're growing a plant, it's fine.
Yeah, I'm with you there. And it also gets me when people are demanding that, their recipe of how they grow is the best and one and only. And I'm like, that shouldn't even be coming out of a cannabis cultivator's mouth. It really depends on what your particular needs are and your resources and your location. And, I would love to grow differently than I do, but I also don't live in California,
Oh, totally fair. Yeah.
Yeah. Which pushes me towards things like short flowering thing and auto flowers, right?
Mm-hmm.
If I lived in California, I would probably still be focusing on my own cultivation on photos. So, all right. So
totally
the next thing I want to talk about is unsulfured molasses. So it is pretty common in living soil for folks to think that they can make up for not giving, I guess I'll call it like whole nutrition, like whole amendments, like let's say like fish meal and crab shell and maybe alfalfa meal, all of these amendments that we love to use in living soil to have an abundant variety of nutrients there for the exudate processors to chew through and then give to the plant. So so the exudates come back and then they feed the microbes, and this is beautiful relationship. And it is not uncommon for people to say, well, I don't need to do all of that to take care of my microbes because I just feed the microbes with unsulfured molasses. And that pains me because I have a gut feeling that just pouring sugar into the soil is not a great idea. And molasses is kind of unidimensional, whereas microbe life is so vastly complex. And I get where they're coming from, right? Because when we are brewing a compost tea, we will often add a bit of unsulfured molasses to that, so that the microbes have something to eat, so that we can incubate them and have a whole bunch of them in a small place that we can then pour onto our plants. So I get where they got the idea, but I think that is a misapplication of the understanding of what effect molasses has on the microbe life in the rhizosphere. So as somebody who has an intimate relationship with exudates, what are your thoughts on adding unsulfured molasses directly into the substrate as part of some sort of nutritive root drench?
I am going to go back to the theme of the day, which is it's complicated and it depends, I think. I think that if you are trying to recover your soil life just with molasses, that it really depends on the health of the soil that you're adding it to, because that molasses, just like it does in compost tea, does have sugar in it. I'm sure things will like to eat what's there and grow, but that means that anything in your soil you're adding it to is going to grow too. So if you've got some nasty, anaerobic bacteria in your soil already, like that's going to love it as much as the next thing does. And the other thing is, like, a lot of the amendments we add provide different different opportunities for different types of microbes to come in. So if you're only feeding molasses and that's how you're getting your microbial life, like, it might get there eventually, but it's also going to be the microbes that specifically like molasses, and you'll miss out maybe on some of the kelp or the crab meal type. And again, this is all kind of conjecture because truthfully, the science isn't there to be able to specifically identify each different bacteria that come with each of these. But that's what my gut feeling would be. If your health of your soil is really good and you're already adding compost teas and things like that, if you're diluting a little bit of molasses in water, I don't think it's harmful. But again, like, I do think that the biggest factor on whether that's a good idea or bad is probably the health of your soil to begin with. Does that make sense?
Yeah, it does. So, is there anything that we can do to encourage healthy exudate creation by the cannabis plant. I mean, we've talked a bit about like just generally keep your soil healthy. Got it. But is there anything that we can do like is there any kind of a fermented plant juice or a supplement or in anything that we could do to encourage exudate creation, because they really do seem to be a baseline to a thriving plant where if you've got poor exudate activity, everything that happens more complex than that is going to be limited by the sad exudate system. So is there anything that we can do that's pro exudate that we should consider?
Ooh, that is a good question. I mean, I think the best answer is probably no and yes. Mm-hmm. No, there isn't a magical thing that, as much as it would be nice to just like promote root health and the healthiest roots you've ever seen just like sprout out of the soil and you're good to go. Because really it actually ends up being about root health. And that's where the yes part comes in, because you can support root health by like the type, like you were talking about earlier, proper watering technique, where you're making sure the roots aren't like super dry and they're not super wet or anything like that. You can promote root health with soil texture. You can add these compost teas and things like that, like we've been talking about, to promote the type of microbes that you want to be there. You can give them lots of space to grow. Obviously, more roots equals more exudates. So if you have a nice big root ball, I'm a big fan of big root balls though, because it's my thing. So that's probably what I would say is like good root health. And so yeah, I think that's that. But I mean, again, it's so hard to say how to get good root health, and it's sort of trial and error. So philosophically speaking, these kind of conversations help too, because you'll find people all over the place that have different ways of making sure they have healthy roots, and maybe one of those ends up working for your particular strain or type of plant.
No surprise that you're a big fan of root balls, being up there in BC, the land of Natasha Riz, who's been on the show talking about root balls as well. My Canadian friends all love the cannabis root ball.
Good roots equals good plants.
Totally. It's so nice to come to an end of an episode without anything new we have to do. You know, usually when we go through these episodes, at the end, there's like, okay, I need to learn about this, and I need to start adding this, and I need to learn how to start, making this Korean natural farming prep. But really, at the end of the story, we've learned a whole ton about exudates today. So A, we stop repeating bro science, and B, we have like a more intimate relationship with an understanding of the biology of our plant. But really, if you're already studying living soil and trying to have a healthy soil, like the exudates will take care of themselves. And it's so delightful to just be able to get new understanding without new tasks.
It's true. Root health is just, you know what? Just keep doing what you're doing and grow the healthiest plants that you can. And the plant is very good at taking care of itself. So if you give it the opportunity to, then you won't have to worry about it.
Fabulous. Well, Sarah, thank you so much for sharing your time and expertise with us today on Shaping Fire. I mentioned this in the introduction to the show that, you really do have a rare specialty, and to find somebody who is, doing this at the PhD research level and also is a home cannabis cultivator makes you exceptionally unique to be able to bridge between the academic science side of exudates and our beloved cannabis plant. And so thank you for, taking 2 hours and sharing it with us so that we can all become more familiar with exudates.
It's been absolutely my pleasure. Thank you so much for having me.
Fabulous. So if you want to keep up with Sarah Lane, the best place to do that is through her Twitter. And her Twitter account is Exudate Rootly. So that's EXUDETTEROOTLY. So that is a great place to keep up on her research and her interests. If you happen to have a more of an elaborate question or comment, perhaps than a Twitter account will allow, she also invites you to send an email if you'd like, and that is slane SLANE, at uvic.ca. So that's UVIC.ca. You can find more episodes of the Shaping Fire podcast and subscribe to the show at shapingfire.com and wherever you get your podcasts. If you enjoyed the show, we'd really appreciate it if you would leave a positive review of the podcast wherever you download. Your review will help others find the show so they can enjoy it too. On the Shaping Fire website, you can also subscribe to the newsletter for insights into the latest cannabis news, exclusive videos, and giveaways. On the Shaping Fire website, you will also find transcripts of today's podcast as well. Be sure to follow on Instagram for all original content not found on the podcast. That's at Shaping Fire and at Shango Los on Instagram. Be sure to check out the Shaping Fire YouTube channel for exclusive interviews, farm tours, and cannabis lectures. Does your company want to reach our national audience of cannabis enthusiasts? Email hotspot@shapingfire.com to find out how. Thanks for listening to Shaping Fire. I've been your host, Shango Los.